So I don't want to take up too much of the time. Our next agenda item is our inaugural mini-seminar by Yaling Liu of Lehigh University. And I will turn over the screen share to you for your talk.
All right, thank you very much, James. Let me share my screen. All right. Are you able to see my screen?
Yeah, but you are.
Let me see what it's.
I think you're sharing in swap presenter view and slide view. So we'll do it.
How about now? It works now?
That's great.
All right, great. Thank you very much. All right, great. It's my honor to start this mini-seminar series. So today I'm going to start with a very smaller scale, talk about some of my recent research on study the binding of SARS-CoV-2 to cell membrane, where we use a multiscale modeling approach on trying to link molecular information to the whole virus and then try to search for some potential drug inhibitors. So I will talk a little bit about background about AC-RBD inhibition strategies, and then we use structure-based screening to search for potential drugs, and then we also try to use multiscale model to link molecular information to the whole virus scale. So first I need to say that I'm not really an expert on this topic at all, because my original research is mostly study nanomedicine design and fabrication and testing. I do both cold screen and diffusive particle dynamics modeling of how to design different coating of such as antibody coating for different types of nanoparticles, and how does this kind of coating will enhance the adhesion and help nanoparticle to penetrate through the cell membrane. So we do both modeling as well as some experimental work where we test and flash those nanoparticles and then quantify their binding to inflammation-induced cell membrane. But since the pandemic, I find there's a lot of similarity between nanoparticle and virus, because if you look about the general like entry pathway of virus, you will see that the virus also have a surface coating, which is those spike protein, and those spike protein will bind with ACE overexpressed on the cell membrane. That will reduce their entry energy and then ramp up on the surface of those membrane, and then you will see the enzyme TPRS2 will cleave the spike protein and eventually activate the virus. So the virus will break the membrane and then enter the cell, and then by into cytosis and then reproduce itself. So I feel there is a lot of similarity between nanoparticles and the virus in terms of the entry process. So so this is definitely a multi-scale problem in the sense that you will see that the whole virus has size about 100 nanometer. Well, if you look about the binding site, which is the spike protein, it is in a scale about tens of nanometer. And if you look about the real binding interaction, which is usually this RBD protein over here, it's in a scale about a few nanometer over here. So that's why we have to use different strategy to model those different scales. Use fluid mist simulation for the RBD protein itself, and then use cold screen molecular dynamics for the virus spike protein. And for the whole virus, we have to use even coarser scale modeling over here. So this is definitely a multi-scale host-pathogen interaction problem. So there's different strategies people usually use to inhibition of RBD and ACE2 binding. So for example, if you look about one smart design people proposed recently is because RBD would bind the ACE2. If you can design some peptide that mimic the structure and the binding functionality of ACE2, then your RBD will preferably bind with your peptide first. So the peptide will occupy this binding site, then your RBD cannot bind with the real ACE expressed on the cell membrane anymore. So this is one of the strategies people use. So obviously there is more general like search for drug, which is actually you can repurpose a lot of those FDA approved drugs or small chemical compound to target the ACE2 and then this RBD interface over here. So you have quite a few targets. Say you can target either RBD or ACE2 or the complex of those two. And then we can usually use so-called structure-based screening to search for drugs and then use molecular docking to find which of those drugs will actually bind best with your target protein. And there is a huge library of FDA-approved drugs or chemical database. You can actually search for a different type of drug and then score or evaluate whether they bind well or they bind bad. So this is the typical searching process. So usually we use the tool, what is called the molecular docking process. So basically you're going to search for a different drug molecule and then different poses of those drug molecules because they're able to rotate and then change their configuration. And then you can actually use different genetic search algorithms to figure out what is the best pose that will fit with those druggable pocket on your target protein. So in general, this kind of docking algorithm will generate like structure that agrees pretty well with the crystal structure people observe in the physiological condition. So what we do over here is that we want to search for some potential drug that will inhibit the binding of ACE2 and this RBD site over here. So we're looking for some druggable pocket, such as this type of pocket that is showing on the, let's say this type of pocket in the middle, right, where you have interface. That's where we care most about, or another drug pocket that is RBD site over here. That's another potential druggable pocket. And then we want to find whether there's a drug that can bind firmly with those pocket on those interface. So the general process is pretty simple. You basically pull out those protein crystal structure from protein data bank, as well as library of drug formula. And then you add hydrogen bond, minimize your structure. And then you use different force field or potential and try to find druggable pocket using molecular docking to find which one is energetically favorable for drug pose. And later on, you can also perform different types of molecular simulation to confirm dynamically it will perform inhibition effect. So the first thing we start with is this popular drug, HCQ. You probably heard a lot about that in the news, so that's where we start. What we find is that it has not very high binding affinity with ACE2, which is this red, which is this green pocket over here. And the docking score we got is about -4 kilocalvin per mole, which is kind of in the low side over here. But we took it and then we perform a steer molecular dynamic simulation to analyze the effect of HCQ on this complex binding. What we do is we pull the RBD from the ACE binding site at a certain rate, and then we attach the RBD with a virtual spring, and then so that we can measure like when you slowly pull it and how large the pulling force is required to separate them. So the reason we want to measure this kind of separation force, because people find from literature, this pulling force is linearly proportional or correlated with binding free energy between two proteins over here. So which means that once you figure out this maximum pulling or separation force, you pretty much get a free binding energy between those two proteins. So this is one of the tests we did. We put the complex together, and then we have a case where we add drug, and another case we don't add the drug. So the drug is docked over here. And then we measure during this separation pulling process, what is the change of force over time. So you will see that the black curve is without drug, and the red one is HCQ docking at this site over here. So we do observe that the force will increase first and then drop, right, which is the separation process. And then with HCQ over here, you will find it that your pulling force does decrease by about 20% or so. So which kind of indicate that this HCQ does reduce your pulling force or inhibit this type of binding. But the problem is that it doesn't bind very firmly, as I said before. The docking score is only about -4. So which means that it doesn't bind that firmly, but once it binds, it seems to be inhibit this binding separation. So we keep moving on, trying to find some drug candidate that will both bind firmly as well as inhibit this kind of binding, or make the metastable complex destable over here. So we target some docking or pocket that is at the interface between the RBD and ACE. So you do see that there is a kind of a pocket that is empty that might be a target point for the drug to fill in over here. So we search out two drugs that we feel that looks promising. One is this compound called SSA09E2. It is actually a drug compound people screened out in 7 years ago for the first round of COVID, not COVID-19, but the previous round of SARS-CoV virus. And then people report this kind of drug might potentially interference with the binding of those two. And another promising candidate we find is a cancer drug, FDA-approved cancer drug called nitinib over here. It shows a promising binding score from the recent literature report, but they didn't actually further check whether the binding of this drug will do inhibition or do nothing. So, but they report that it has potentially a good binding score over here. So we take from here, and then we first study the binding of this SSA09 with both RBD and ACE2 as well as the complex. So what you can find over here is that the binding score of either RBD or the AC is not very high, -5, which is kind of reasonable. But the binding score at the complex pocket site is really high, which is -8. So which kind of indicate that this drug will likely bind with the complex rather than individual of those two. So it's kind of an interesting finding over here. And the same happens to actually for this cancer drug, nitinib over here. It has pretty high binding score of -8.32 on this docking site at the complex over here, which is very similar to the SSA, but they kind of bind as two upper side. One is at the front side, another one at the back end over here, both at a similar interface, but different side of this pocket. So the next thing we study is we study the root mean square dynamics of both drugs. So this one is for the ligand itself, which means that we want to see whether this drug will bind firmly. So you will see this SSA bind pretty firmly. So the root mean square displacement of this drug kind of very stable. It doesn't fluctuate. Well, the other one, the nitinib, has a lot more fluctuation, which kind of means that their docking is not very, very stable. It's kind of bind and then off, on and off. So it's not as strong as SAA. So if you check about the root mean square displacement of the complex, RBD-ACE2 complex, we observe similar trend. So the black one as well, the red one is the baseline. You will see that this is without any drug, which is the red curve over here. It kind of locally fluctuate. But once you add in this drug SAA, you will find it that the root mean square displacement will have much larger fluctuation, which kind of indicate that the overall complex structure is not stable. It's kind of fluctuate up and down or on and off frequently. And if you look about the particular residue that is triggering to this fluctuation, we can find it that majority of those fluctuation of the residue is at the beginning part, which is from sequence from 300 to about 400, which is actually located at the interaction between the ACE2 and RBD over here, which kind of indicate it's the contact interface between those two proteins is fluctuating a lot and become unstable under this drug SSA over here. So nitinib also have a little bit fluctuation, but not as much as SSA protein over here. So if you look about the molecular dynamic simulation that tells more detail, you will find it that the SSA has very good docking. It's kind of bind very firmly over there and then make the overall structure fluctuate and unstable. while this nip over here, it's kind of partial stable, right? You will see that they fluctuate and part of it is anchored over there, but it has another end that is dangling around. So which explains why it doesn't actually bind that firmly or doesn't actually inhibit that well over here. So from this one, we find it that this SAA might be a promising drug that both bind firmly and also make this complex structure unstable once it's binded over there. So of course, in order to confirm this, you also want to check about what's going to happen at the whole virus scale, because what we study or majority of the drug screening is only focused on a small portion, right? One single pair of RBD and ACE2. But as you realize that the cell membrane, we might have multiple of those ACE protein that bind with multiple of those spike proteins over here as well. So that's why we're also interested to see how does this kind of RBD AC binding will influence the overall uptake or entry of this whole virus over here, which is the virus uptake process. So what we do over here is that we use a co-screening approach, because obviously with millions or more than millions of atoms, it's impossible to study use optimistic simulation. So what we do is that we co-screen, grouping different groups of atoms together into those beads. So we can actually simplify each of those RBD and ACE with hundreds of or thousands of beads rather than millions of atoms over here. So we use what is called shape-based mapping to map all missed structure into those co-screen beads over here. But the key thing is that we need to develop a proper co-screen force field. And the requirement over here is that we want to preserve the flexibility or the dominant mode of those moving spike proteins, because recently there is quite a few papers reporting that the spike protein itself is not rigid. It is actually swirling around or rotating around, and there's an average angle. So it's a very flexible structure. Such flexible structure will also help the spike protein or the virus to actively capture or encounter with those ACE protein in an active way. So we actually want to preserve the feature of this flexibility over here. And we also want to preserve the interaction between those proteins as well as the mechanical properties of the whole virus. So this is still ongoing work. We're still developing the potential. But if we're able to figure out a proper potential, we can develop this kind of whole virus scale co-screen model, right? We have all those distribution of those spike protein and then preserve its mechanical properties as well as its binding with ACE side over here. So for this binding part, again, we want to actually preserve the binding free energy. If we use a very simple linear-to-noise potential, then we need to tune the potential parameters so that the overall binding energy will be equal with the binding energy obtained from all optimistic simulation over here. So we're trying to preserve the binding energy of both internally as well as between RBD and ACE protein over here. So once you obtain that kind of binding energy or co-screen model, then you can actually run steer molecular simulation or umbrella sampling to sample how large the force is needed to push the virus into the host cell surface, or how large the uptake force or uptake energy is supposed to be. So you can actually plot out the free ramping energy as a function of engulfment depth, which means that as a deeper and deeper the cell engulf into the cell membrane, we can actually plot out the change of the total energy. So if we say the binding energy or binding energy surface is called A, you will find that when the binding energy is very small, it's hard to uptake or hard to enter, right? The total energy will decrease and increase back. But if you have higher and higher binding energy between RBD and ACE2, you will find it that it becomes much easier and easier to uptake, which means that it's more favorable for the virus to enter the whole membrane when you have stronger and stronger or more and more those binding sites over there. So of course, another thing is that how did you know your model is correct, or how did you validate the model? So I'm working with one of my colleagues at Lehigh University, Frank Zhang, who was working on AFM measurement of this RBD binding. So what they do is that they can conjugate RBD on an FM tip, and then they can coat a layer of AC receptor on a cover glass, and then they will put this FM tip close to the surface and then pull it. So you will see that they're able to measure the unbinding or the pulling force at different pulling rate over here. And then you can also check out when you have blocking or inhibiting drug over here, how does this kind of blocking or inhibiting drug will reduce this kind of a separation force. So you can actually use those type of molecular scale validation data to validate the model if you want to verify your model is correct. So for the last two slides, James asked me to share about some potential direction that we can collaborate, as well as whether there is some interscale connection. So one collaboration opportunity I'm looking for is trying to use data-driven approach to develop some coarse-grained force field between ACE2 and RBD. As I told you, we haven't really figured out an accurate or good way to derive this type of coarse-grained potential. As you can imagine, you have tons of atom group you need to group together, and each of those group you actually need to derive their coarse-grained potential over here. So that's why in the literature, people actually use machine learning data-driven approach, because we have a lot of data from optimistic scale already. And how do we feed those data properly to derive those potential in a data-driven approach, so that we can actually derive in a fast and accurate and preserve our flexibility for this pair of potential, both for the coarse-grained approach as well as for the whole wire scale. I hope that some people who's familiar with machine learning or data-driven or potential development could collaborate on this type of topic. So another one is for connection between different scales. So obviously, my work is mostly focused on molecular scale, interaction between spike protein ACE2 to the whole virus. But I believe that there is a lot of room at larger scale. For example, the AC expression in organ scale might be heterogeneous, right? Depends on different location or different organ. You might have totally different AC expression density, and then that will also trigger different uptake process. And then maybe we can also couple with some larger scale mathematical infection models. I believe this is some work from James' group, where they studied the spreading of those virus, but coupling with this uptake process might help the spreading modeling as part of the input over there. So other topic could be that we can also study other type of protein interaction, or the fusion of the virus, or the overall ramping process. So I'm looking forward for discussion and collaboration with people in this working group. Thank you very much.
Thank you very much. That was great. Thank you for leading off. Appreciate it. We have time for just a couple of questions, if there are any from the audience. Jacob.
So you're trying to connect, you're working at the virus level, or like molecule level, eventually to the cell level. This is the levels, and you're trying to reach higher up to, let's say, you eventually want to do a spreading of disease, so probably up to the population level. What ideas you have to put in the middle? What other ideas you have to reach up to that level?
That's a good question. Frankly, my immediate answer is no, I don't know how to do it. I feel this is definitely not a concurrent model, right? This is more like a hierarchical model. You derive from molecular scale, get this binding affinity, and then you will got, I would say, maybe the probability of entry and uptake, and then that will link to maybe the density of virus at local point. And then maybe you can link it to the distribution of ACE2, and that will actually couple to the organ scale. So I believe it's more like a hierarchical coupling rather than concurrent coupling, more serving as input of the next level.
Okay, I'll try to rephrase my question. Maybe I'll get a clearer answer, because it's hard for me at that level that you're talking. Please help me. So okay. I'm trying to see if I can get a characteristics, let's say, of an individual, because you have those nice molecules, and you can compute some sort of probability that it will attach or not attach to the cell. So far, I'm correct?
Yeah, that's right. It's a molecular scale or a single virus.
Good. So if you have a single virus, and let's say you can compute from a single virus its ability to attach to a cell. Let's say you have other properties of a human being. Let's say height, weight, number of cells, I don't know, something, or number of susceptible cells. Can you give a probability of the virus attaching to that human and causing the disease? Is it something that is like foreseeable, or what things do you need to actually compute that?
That's a pretty complex question, I would say. First of all, you will see that in order for, the first thing you need to say is how many virus will really enter, right, or contact with the cell.
Okay.
And the question I'm addressing is that after the contact with the cell, what is the chance or what is the probability it will enter the cell? After the enter cell, then the next thing is how many of them will produce themselves and then spread out. So there's multiple steps. I think what I'm addressing over here is one of the intermediate steps. Is if a virus is contact over here, what is the chance it's going to enter?
Good. So you already have some of the intermediates. If we can figure out some of the other ones, we can rise up to the population level. And the question is, who's in this group is able to do those other intermediate steps?
I think there are people in the group who are interested in barrier function. There are people in the group who are interested in airway transport. There are people in the group looking at ciliary transport and mucus. And so I think there will be, as time goes on, we'll see people filling in many of these pieces. And I think it's great that you bring up some of the issues that would have to be addressed if you wanted to have a whole sneeze to infection model. There's a lot that would be required. Is there time? We have time for one other very short question. I'm afraid that we have to stay on a tight schedule to keep it a mini seminar. So if there's one more question, you're welcome.
So yeah, if I can ask a quick question. I'm actually interested in particle that could be viral particle transport in the airways. But so to bind to these AC2 receptors, do we have in the group expertise in what's the distribution? Where are those AC2 receptors located in the lung? Are they mainly in the airways? Are they mainly in the gas exchange region? Because that's going to affect the distribution. The particle deposit non-uniformly in the lung. So that's an important aspect also.
Well, certainly different cell types have very different levels of expression of ACE2. So, and even within a single cell type, I'm sure there's a significant difference.
Yeah, so that's probably something that will need to be looked at.
Yeah, that's definitely some mission information because we do actually search in literature about this critical information as you just asked, and we didn't actually find much clear data on that, in particular quantitative data. There's only like some description, like some people find like from the tissue sample where there is ACE2 overexpressed, but there's no quantitative data about the distribution among different like locations.
So that would be a good point to raise in the discussion groups, that that's missing information,
Yep.
Critical missing information. Okay, any other quick, I hope people can continue this discussion offline through the Slack and elsewhere. Thank you again, Yaling. That was wonderful first talk.
All right, thanks, James.
If you could, I'm going to go back to our less interesting business talk now.